Good afternoon, everyone. Let me start by thanking Frank, Michael, Forever Healthy for organizing this meeting. It's actually really exciting to be here. And I'm a little nervous, to be honest, because I'm going to be talking about data and not maybe what was expected. But I think we have new results that I think will be of interest to the field, and I'm really excited about sharing them with you. So we've come a long way in this field. We're about 20 to 25 years in what I and others have called a revolution in the science of aging, which means that aging has become a scientific discipline that can be studied using the tools of molecular biology and genetics and animal models and so on. And we've learned quite a bit in the last 20 to 25 years, including the identification of what I would call dials, nodes in aging pathways that we can trigger, that we can modulate, and by which we can change the rate of aging. So all of this has obviously incredible potential. And one typical example that many of you are probably familiar with is the idea of rapamycin, which is identified as a drug that targets a specific protein which is conserved across all species, called TOR, target of rapamycin. And animals treated with this drug actually show an increase in lifespan, as shown here. Now, the amazing thing is that rapamycin is actually present in the clinic. It's used at high concentration for immunosuppression, and there lies the potential of using it as a therapy against aging. But this is not the only one. We now have over 100 interventions in mice that can extend their health span, and sometimes quite significantly. A number of these molecules are in the clinic, and therefore we can expect for the next 20 years a flourishing of clinical trials and new data showing for the first clinical trials targeting aging. I've listed a few here on this slide. You'll see metformin, acarbose, again, rapamycin are 3 drugs that are presently in the clinic. We're talking about senescent cell clearance. Their first clinical trials actually are being conducted with those, FGF21, growth hormone, releasing hormone, and the list goes on and on. So obviously an enormous amount of excitement in the field, but also a gigantic problem, which is how do you conduct clinical trials against aging? How does one do this? There are regulatory problems, but there are also practical problems. No one is going to be doing clinical trials for 20 or 30 years. Hence the need for a whole field called the biomarkers of aging. And this is what I'm going to be telling you about. When we think about biomarkers of aging, and I'm thinking here about a universal biomarker of aging, they should have a number of key characteristics that I've listed here. First, they should be a reflection of a ubiquitous aging-related process. They should be universal, working across different tissues. They should be based on a cellular and subcellular level process. They should be predictive of health span and lifespan, should vary in response to intervention, and they should work in men, women, and different ethnicities. The short answer is that we don't have these markers right now. The field is still searching for really what is going to be emerging as the best marker. But there's been some progress, so I don't want to make it sound like we haven't made any progress. And right now, one area that has generated probably the largest amount of interest is the idea of these epigenetic clocks. And this graph that I've taken from a recent review shows 2 variables, accuracy and utility. Obviously, in terms of utility, blood tests are easier to do, but in terms of practicality and the precision of these assays, DNA methylation has emerged as the largest and probably the most important. And this is what I'm going to be discussing today. So what are these DNA methylation clock, or so-called epigenetics clock? I just want to highlight very simply what the basis of their generation is. You start from, let's say you start from a number, the way you generate it, you start from a large cohort of individuals of different ages. They all have different chronological ages. Some of these clocks actually have incorporated clinical characteristics such as blood markers. And one example of this is the pheno-age clock. Then these clocks are actually regressed onto using machine learning and a variety of different paradigms. Either the 2 biggest ones are so-called lasso and elastic net. You can regress these variable chronological age or clinical characteristics onto the amount of methylation at a number of CPG sites in the genome. Just one word back also, what are these CPG sites? This is a dinucleotide, CG, You all know about ACTG in the universal DNA code. There are about 20 million CG letters in our genome, and about, I don't remember the exact number, but I think probably about 10% of those are actually methylated within the genomes. And the level of each of these methylation can change during aging. So the idea is that you do the regression using machine learning. This generates an algorithm which will select the number of these methylation markers. And you can take a sample which is not the original samples, but another one, a person that comes into the clinic. You measure the DNA methylation level at each of its CPGs, and then you push this back, and it actually generates a chronological age. And what turns out to happen is the age that you get can fall perfectly on the line, but it can fall above or below. If it falls above the line, that means your DNA methylation status is more akin to someone that should be older. If it's lower, it corresponds to someone who is less old. And therefore, there's been the idea that you can actually predict what is the trajectory of aging in individual people. Now, the field has been incredibly busy, and we now have many of these methylation clocks. And Steve Horvath has been the true pioneer in this field. He's generated several of them, some that are across different tissues, some that are specific for the blood or skin. Hannum has generated their own clocks, PhenoAge by Morgan Levine, GrimAge by Horvath again. And so there's a lot of progress. There's been a lot of data showing that these clocks actually fluctuate, not only as a function of disease, as predictor of disease, but also in some cases, some claims have been made that one could see reversion of aging. There have been 2 clinical trials now in humans in which it's been claimed that there was a reversion of aging. And what I will show you today is I think I have strong doubts as to whether these data actually truly demonstrate a reversion of aging. Now, the reason we got into this whole thing is many of these clocks are measured by taking a sample of blood, in the peripheral blood. And my laboratory studies the immunology of human aging, and I've always been bothered by something. And this is the fact that blood is an incredibly complex compartment. There are more than 60 different cell types in the blood, and you can divide them. All of them are derived from a so-called hematopoietic stem cell that you can see right at the top. And then they divide into 2 broad kingdoms, the myeloid progenitors and the lymphoid progenitors. The lymphoid progenitors lead to the T cells and the natural killer cells and the B cells. And this one goes to all of the other cells that you're probably familiar with, basophils, neutrophils, eosinophils, and so on. This is only a very small picture of the complexity. The area that we study the most corresponds to these T cells. Now, I'm going to tell you a few words about the T cells and why this is important in terms of its relevance to aging. The T cells have a unique developmental pathway. In a way, they start in the bone marrow, the progenitor goes into an organ called the thymus. Again, the thymus is one of the earliest organ to show aging. By the time most of us are 50, your thymus has degenerated to almost non-existence, which means that at that time you rely only on what is present in the periphery. So what happens in the thymus? These thymocytes generate these naive CD8 T cells and CD4 T cells. I'm only going to talk about CD8. Everything that I'm going to tell you today about CD8 applies to the CD4, so it's just a way of simplifying the presentation. So these naive T cells then migrate, and when they recognize their own signature molecule, be it a virus, a bacteria, or any kind of pathogen, they get activated, they start dividing, and they become what's called effector and memory T cells. And this is the basis for vaccination. When you receive your COVID-19 vaccine, it is recognized by specific naive T cells, which recognize it, they start proliferating, and then eventually they contract, and they leave a memory T cell, so that when the true COVID comes in, or you get revaccinated, you get an amplification of these cells. So this is the basis of vaccination. Now, we know vaccination decreases very strongly during aging because the pool of your naive T cells decreases. And why does it decrease? It decreases because your thymus is no longer there. So as we think about rejuvenation, Thymic rejuvenation in the long run is going to be a critically important area to study. And by the way, it is very little studied. So if you are if you're a student or a postdoc looking for an exciting project, definitely is one area that I would investigate in. So that being said, the first thing I'm going to show you in terms of data was asking the question, what happens to all of these cell types during aging? And the way we did this is by working only in humans again. We used a cohort in this case. This was about 45 humans, males and females of different ages from 20 to 82. And we got blood from them, stained, and this is the key technological advance here that I want to highlight, is the field of flow cytometry is a field that allows you to label all of these cell types, these 60 cell types, and to enumerate them in the blood of anybody. And so there's a technology called the Cytec Aurora, which we purchased a couple of years ago, and it's allowed us to do staining with 38 different color panels. Now imagine the complexity of a staining with 38 different colors. Traditional flow cytometry takes about 5 to 6 colors. So this has enabled us a very deep so-called immunophenotyping of blood cells during aging. And this is what we found. So we got a lot of data with this. We apply something called a dimensionality reduction test. In this case, it's called UMAP. There are a number of these tests that allow you to take a data set with 38 dimensions and to reduce it to 2, essentially. And so this allows you to detect pretty much every cell blood type that you can analyze, and then to look at their relative abundance in our cohort of different age. And what we saw is that the NK cells increase, the monocyte increases. By the way, there's nothing new there. People have known for a long time that when you age, there's a shift to myeloid cells, like monocytes, and a decrease in T cells because they're dependent on the thymus. As shown here, the CD8 T cells, you can see them decreasing. The gamma delta T cells, another subset of T cells. Nothing new there, but still quite interesting to validate it. Where it got really interesting is when we started looking at specific T cells. Now again, we can do the same dimensionality reduction just on CD8 T cells, and you can see again the complexity on the left panel of all of these different types of T cells. Now we can ask for each of these populations, how do they vary during aging? And this is what we saw. Look at the fraction of naive T cells during aging, an incredible steep decrease, close to 0 by the time you're 80 to 90, which corresponds to what we know. If you vaccinate someone who's 70 years old, the probability of your vaccine working is actually 30%. Think about this number, 30%. This is an incredible number. Now, the naive T cells decrease. Central memory, there are 2 different populations. You can see them increasing. And that corresponds to the shift that I've told you about, this shift from naive to more differentiated cells. Now, when you see things like this, and you realize that people are doing epigenetic clocks on peripheral blood, the big worry for me was, are we actually looking at true change of epigenetic marks in cells that are aging, or are we looking at shifts in populations? Imagine that some of these populations, imagine that the naive T cells look younger. As you age, their fraction decreases, therefore the whole blood is going to look older because the fraction of the cells that are younger is decreasing. And so that was the question that we asked. Very simple question. Can we actually measure the epigenetic age of these individual populations in any given individuals? Are they different? Are they the same? Are they congruent, or are they different? And this is where we got our first surprise. So the first experiment I'm sharing with you is ask a question: what is the impact of CD8 T cell differentiation on epigenetic age? And we did this in 7 donors who were between 30 and 77 years old. And we used, again, the flow cytometry that I've showed you in combination with something else called sorting. You can not only enumerate the cells, but you can also separate them. So we can, using the flow sorter, we can purify the CD8 naive T cells, central memory, effector memory, and a last population, which is terminally differentiated, so-called AMRA. Then we took these cells, extracted DNA, and measured the methylation state. And we did this, by the way, in collaboration with the Clock Foundation, which was started by Steve Horvath. And what we saw was actually stunning. This is probably the most important slide of the presentation that I want you to remember when you see a clock. Because what we saw is that any given individual, this is the average of the whole population here, in any given individual, the naive T cells are close to 20 years younger than the effector memory and the terminally differentiated cells. As you can see, so this is, again, I hope everyone is following me still. That means that in a given individual, you can see cells that are 20 to 25 years younger than other cells. So what really does this mean? So this really was the first symptom or a sign that we had to worry about what these global clocks conducted on whole peripheral blood actually truly mean. What was even more interesting is that this was not the whole story. So differentiation is not the only thing that leads to a change of the clock. The second thing that we saw, if you look at all of the naive T cells, they're shown here in red or orange, you can see them here, actually in different individuals of different ages still saw the clock moving. And so what this really, and the central memory, the clock was moving a little slower, and the effector memory even a little slower. So what this, the conclusion from this, and this is the second most important slide, is that the clocks represent a mixture of CPGs, the dinucleotides, that vary across distinct differentiation states, and so this is the first part that I told you, and of CPG that change as a consequence of aging. So all of the clocks, by the way, everything that I've told you about the Horvath clock we've seen in the Hannum clock, this is not unique to the Horvath clock. That means these clocks that we're looking at right now are a mixture of two variables. And as a scientist, I know that if I'm studying a process that's depending on two variables, looking at the end results is very complicated. You can't really sort out what's happening. And so this suggests, again, that change in differentiation could also give the illusion of aging acceleration and aging reversal. And I think we have evidence of both cases. So this data basically brought to our attention the fact that there is a need to separate these two variables, the differentiation versus the true age process, and to generate a new clock that measures aging exclusively. And this is what we have done. So this is the work of a graduate student, Alan Tumusiak in the lab, who joined a little more than a year ago. It's just been amazing to see him doing all of this work. So I'm not going to go through the technical details, but I want to explain a little bit of how we went about doing this. So we started by creating a very large database from existing literature. All of this is available online. You can download it. And this database had 400,000 CPGs, 14,000 samples, and 61 data sets. We did some filtering for a number of criteria. This left us with 350,000 CPGs. And Alan did the critical thing, which is to remove all of the CPGs that were linked to differentiation. Remember the ones that we saw initially. Remove them out of the clock. And so this left us with 84,000 CPGs. And again, we use this now to create two data sets, a training set and a test set. The training sets essentially allowed us to do exactly what I showed a few slides away. Look at the age, look at the DNA methylation, use a regression analysis, and identify a series of sites that are actually building a clock. And this is what it looks like. So the prediction would be that a successful new clock should look this way. It should still vary with age, as we had predicted. But when we look at the different T cell subset, it should actually be flat. It would tell you that this clock now is looking purely at age and not at differentiation. And this is what our new clock is looking like. First, we took the prediction data and the algorithm and tested it on the data set that we actually had used to generate the data. So that's really the lowest threshold to actually make sure that your clock is working. And you can see that it works actually beautifully. This is the age of the individuals. This is the predicted age using our algorithm. You know, a correlation coefficient of 0.97 doesn't get much better than this, and you still have some variation around it, which could be an indication of different people aging at different rates. Now, second step was to take the new algorithm and apply it onto a test data, and this is what we saw. Again, you can see now the spread is a little wider, but the correlation coefficient is still greater than 0.91, and the clock really is able to predict all ages. If we do the same thing on blood, only on blood data, you can see even though this is the compartment in which we would expect to be the largest amount of artifact, you can see that the clock is still behaving remarkably well. Now, when we do this also on the purified CD8 T cell populations, on the left panel is what I had showed you we did earlier. Remember the naive central memory, effector memory, TEMRA? You can see them aging very rapidly. Again, these are individuals. I had showed you earlier the average. You can see that in every single case, we see an acceleration of aging as a function of differentiation. When we do our new clock, this is completely suppressed. So we think that the clock behaves in a way that we were expecting it to behave. So time then to have a little fun with it and try to see, okay, what does this really mean in terms of biology? Again, another way we did this is to compare, because you might say, well, this is what's really unique to the Hannum clock or to the Horvath clock. We actually compared all of them. And we compared this to yet another data set of purified, sorted CD8 T cells from another group. And we compared, again, the predicted age and the measured age. Now, all of these clocks, again, are underestimating, and our clock falls right on this is only in the naive T cells, and our clock, again, finds exactly the same age. So demonstrating that our clock is insensitive to differentiation state. The same thing you can do. One important aspect that worried us is that, remember when we subtracted all of these CpGs that were CD8 T cells, this was done with CD8 specific T cells. So the worry was that maybe we're looking at something that's specific for CD8. So what about if we take this algorithm and use it on CD4 T cells? It still works exactly the same way. You see the other clocks actually finding a delta, sometimes 30 years. Our clock falls right on the line. So our clock is truly insensitive to this T cell differentiation. And we have preliminary data that I don't have here, but that shows that this works across almost every single tissue and organism, which almost seems miraculous to me, frankly. But that's the data. Now, why is this important? Here's why this is important. You might have seen some papers claiming that COVID-19 is associated with aging acceleration. And actually, there have been a number of papers published claiming this. This is one of them. And the data is using different clocks. Actually, you find different things. Some age acceleration, some age deceleration, and so on. Our clock is insensitive to this. Now, why would COVID yield a change in epigenetic clock acceleration or deceleration? Because it's associated with immune proliferation and a massive shift from naive T cells to differentiated T cells. So again, you have the illusion that this is aging, but this is not aging. It's actually a change in the composition of the immune system linked to immune activation. Now, here's another one that I think made me quite happy. We took our clock and said, okay, what happens when we actually reprogram cell? Many of you have heard, I suspect, about the whole idea of rejuvenation using Yamanaka factors. These are factors that are able to take a terminally differentiated cell and to bring it back to an embryonic stem cell. And other clocks have shown this. Have shown that when you do this, the clock actually rewinds, and we see exactly the same thing. Here's an average 40-year-old population. You can see as the cells become terminally de-differentiated, the clock winds down to almost zero. So this is really a good example that whatever we're measuring is linked to the aging process. Now, because of the time, I only have five more minutes. I won't be able to go through all of the analysis that we've done, but we went to the next step, which is to look, what about all of these papers that claim that there is aging acceleration under a variety of clinical conditions? What is happening with our clock? And here's what we found. The summary is that many of the things that have been reported, at least in the literature, as being linked to age acceleration, we do not find them to change using this new clock. Now, if you look at them, many of them, obesity, smoking, Alzheimer's, rheumatoid arthritis, multiple sclerosis, celiac disease, COVID-19, HIV, depression, many of those are so-called chronic inflammatory states, and under conditions which you would expect to see a shift from naive to more terminally differentiated. So immune aging is a manifestation of aging. So I'm not trying to say, so what happens is one reason why the previous clocks work so strongly is because they take two variables of aging. The shift to immune activation is part of aging. We extract this variable out of the whole process. So what we have found is also linked, is actually truly strongly linked, Parkinson's disease, dementia, Huntington, and many forms of cancer. And this is quite interesting. We don't really understand what is the link here to cancer. And there's a lot more work that needs to be done. Now, a couple more facts that I want to share with you is the fact that our clock shares very few, and this has been a problem in the field, is that when you look at the CPG that are identified by the different clocks, there's extremely little overlap. Even though they all measure aging, the question is what exactly is the biology? And that the field has been struggling with the idea that we have CPG that vary, we expect them to induce change in gene expression. Why are the genes that we're looking at not speaking to us in terms of aging? And I think one of the reasons we think is because this is an admixture of various sites, some that are linked to differentiation, some that are truly linked to aging. And so our new clock is comprised of 518 unique CPG, and I can tell you without really going into the details that when you look at them, they speak a lot more to the aging pathways that we know than the traditional clocks. And I think this is an area that we're going to be exploring actively. So I want to finish with a summary of what I've just told you. And hopefully if you've lost me halfway, if I've lost you halfway, you'll be able to catch up. So CD8 T-cell differentiation state plays a major role in epigenetic clock age. I think this is really one really firm data that we can state. We've created a new clock which is resilient to changes in T-cell differentiation with interesting properties. We are currently investigating the connection between epigenetic age. The bottom line is that even though we have a new clock, we don't know what it's really measuring. We think it's age. It's very strongly linked with age, but we don't know what it is measuring. And so we're trying to correlate all of this with measures of health span, transcription, and epigenetic changes. We also believe that as a field, if you have a wrong assay to measure your most important variable, and I'm not here to stand that I have all of the answers because we don't, but I think our data should raise a number of key questions as to how we use these clocks and what their utilization means to the field. So I'm calling for an interpretation of age acceleration, age rejuvenation, to be reevaluated based on these data. And so the final one, the one that actually excites me the most, and one reason I wanted to present this to this group is, what is our clock measuring? I mentioned there are some genes and pathways that are emerging when you look at the CPG sites that are speaking to more traditional aging pathways. So that's important. And there are also novel targets that we think could be novel targets for interventions. One area that I'm excited is the idea also now we have a screen that we can utilize for age reversion in vitro. You can do this actually in the vitro setting. And one of the ideas that we're exploring is the creation of a startup that would actually use our assay and either approved or a number of small molecules for their ability to actually move this clock. And so I'm quite excited by this new direction that we're taking. I want to finish with the most important thing, which is I'm just here talking about this work. It was all done by incredibly brilliant group of individuals in my lab. Alan Tomusiak, who's a graduate student, Herb Kassler, who's been working with me for 20 years on immunology, Hiroyuki Matsui, and Marlene Cervantes, who are two great postdocs from Japan and actually Los Angeles. I also want to acknowledge the critical support of two organizations that have actually made all of this work possible, Forever Healthy. As part of the we have a new project with them in which the flow cytometry data that I've showed you has been supported with them. We've also been supported by Michael Antonov and his foundation. And I want to express my gratitude to both and all of them for our support of our work. And I'm finishing 12 seconds in advance. I want to thank you for your attention, and I look forward to our discussion.